Hello, greeting of the day. I am Dr. Sachin Tyagi from Life Science. So here we discuss the very important topic of the microbiology, which is sterilization techniques and control of microorganisms. Yes, can we control microbes? Yes, we can do. We can do by the several kinds of agents which termed as a biocides. So biocides is a terms we generally referred for the agents which can control by these physical agents, chemical agents and mechanical and biological agents. Physical agents are heat, radiation, etc. And chemical agents are those agents which are chemical in nature, and general examples are phenols, alcohol, and other. Whereas mechanically we can filter, like in filtration technique we can filter by the microbes. And we can also control the microbes through the biological agents like vectors, etc. So these control of microorganism generally refer these four different terms. The first term is sterilization. Sterilization is a process by which when we remove all the contaminants from the surface or inanimate objects, while the disinfections having lower magnitudes, this used when we reduce the microbial counts or the harmful microbes from the surface or inanimate objects, while the sanitization and antisepsis are generally the alternative terms of both of these terms. And the meaning of these antisepsis and sterilization are when we reduce the microbial loads from the living organism or the live cells. So if we see that the graph of the agents, that sterilization are totally remove or lower the microbial population in a very short times, while disinfections usually take this some times. And in terms of antisepsis, which is a lower magnitude, so it take some times. So if we are talking about the agents, first methods by which we can achieve the or we can control the microbes is physical methods. And the common example of the physical methods or common route by which we can control the microbes are the heat. So heat generally used from the ancient times, and in microbial respect it is a two types. Number first is moist heat, and second one is dry heat. So, if we use the moist heat, we need a special instrument in microbiology respect, which is known as we are very familiar of this instrument, which is known as autoclave. And if we use several oven, when we directly apply the heat over the several glasswares or other objects, so we can use the oven over here. So, what is the principle of these moist and dry heat? So, in moist heat, generally water has been heated at temperature of 121 degree centigrade, and this temperature can be achieved under a pressurized vacuum systems, and pressure we readily apply over here is 15 atmospheric pressure. So, when the water vapor convert or the in humid conditions generally enter inside the microbial cells, and coagulate their proteins. So, there are many types of protein which are responsible for the microbes survival. So, they coagulate and lose their functions, and ultimately the microbes dies occur. So, welcome back to our control of microbes. So, in previous lecture we were discussing about the control of microbes by the physical methods, and the first method or the agents by which we can control the microbe is heat. So, heats is a two types, which is moist heat and dry heat, and autoclave is a techniques by which the moist heat is used to control the microbes. The this autoclave, the first discovered by the Charles Chamberlain, Chamberlain in 1884. So, after the discovery of this autoclave, the revolution has been taken in microbiology field, and we can culture the microbes in a controlled environments where we can avoid the contaminations or unwanted microbes, and we can grow the desirable microbes in laboratory. So, basically what happened in the autoclave? So, it is a closed chambers where we having a temperature probe, or this temperature probe detect the temperature of these closed systems, and we having a pressure probe So, in this autoclave apparatus, we need to control, or we need to examine the two things. One is temperatures and second one is the pressures. So, usually the pressures at which the autoclave has been achieved is 15 psi, and temperature is usually 121 degrees centigrade. So, why we use or why we take this 121 degrees centigrade and 15 psi pressures? Because this temperature cannot be achieved in a pressurized system or this pressure. So, when we applied this pressure systems, this temperature can be achieved. As you all know that water is boiling the start above the 100 degrees centigrades. So, when we apply these pressures 15, so we can achieve this temperature. And why we achieve this temperature? Because we need this temperature, because we all know that all vegetative cells has been killed around 70 degrees centigrade. So, we all know that the upper temperature limits of the vegetative cells is usually 70 to 72 degrees centigrades. We all know that there is another phase of the bacterial cells which is known as spores. And these spores, which is the resisting cells, or the which type of cells which can resist the high temperatures, and the spores has been killed at 15 to 20 minutes at 110 to 120 degrees centigrades. So, we need to proper sterilize conditions, or the sterilization, we need to achieve this temperature. That is why we required about 121 degrees centigrade temperature for this autoclave, and this can be only achieved by when we apply these pressures, because we need to kill the spores. Fine. Third thing, we need to consider the time of the autoclave. The time is usually for 20 to 30 degrees, 30 minutes. So, this autoclave, another factor which is used, which is how we can conclude it that in autoclave, sterilization has been taken place. So, for this confirmation, we need to add some bacterial spores, and these spores is known as Geobacillus stereothermo. So, these organisms or the spores of these hyperthermophilic organisms or the thermophilic organism has been put inside the vessels of the autoclaves, and after autoclave, in this autoclave we along with the another material, we also put these spores in a test tubes. So, when we subculture after autoclaving procedures in a petri plates, if the growth has been not appeared, after subculturing, if the no growth has been taken place in this petri plate, so we can confirm that there is a proper sterilization has been taken place by this autoclaving methods. So another heat types is pasteurization. So there are some type of products like milk or the liquids. Basically this technique applied on the liquid, which is heat labile. Heat labiles means those liquid which change their texture, their color or their properties after heatings. So we use these pasteurization techniques, and these techniques or these methods has been using the control temperature, control specific temperature. So at this temperature the liquid which is heat labile does not lose its properties, its textures and its properties. So usually it is a specific temperature. So this is vary between 55 to 60 degrees centigrades. Usually in the process of milk, when we consume the packaged milk, which is usually sterilized or by the this pasteurization methods. Now we are moving to the physical or mechanical methods, or sometimes it is also referred as a physical methods previously, but now it is separate sections, mechanical methods. And which is of filtration. Filtration or the sieving. So in these filtration methods, when we can apply heat on the certain type of agents which can destroy by the heat, so we can apply the filtration methods. So filtration has been a filter has been a three types. One is a depth filter, second one is membrane filter, and so basically these are the two types, depth filter and membrane filters. So what is happening in the depth filter? So this depth filter has been made up of by the certain kind of agents like diatomous earth, asbestos, and some kinds of materials. So the name of this depth filter, because due to this materials we create a depth So this is made up to this is the depth or the width of the filter, which is made up by this material, and when we put our liquid solutions over here, and when we applied the pressors or the suck towards the insides, so the some kind of cells or microbial cells has been trapped over here, and only the liquids which is the microbes free has been achieved by these depth filters. Second type of filter is membrane filter. As the name suggests, this filter made up of the certain kinds of membranes, usually by the cellulose acetate, cellulose nitrate, and polycarbonate, polyvinyl fluoride. So usually this is called the cellulose membranes or other membrane which is made up of by these materials. So by these membranes we can filter the liquid as well as the air also. We all know that in current COVID pandemic we generally use the N95 mask. So this N95 mask is made up of by this one kind of materials and make a membranes which filter the 95% of the microbes or the substances or the air . 95% substances of air has been filtered by the N95 mask, because it is filtered the 95%, that's why this mask has been named after this N95. And the pore size, usually the pore size is about 3 micrometer. The pore size of this membrane is 0.3 micrometers. Because this is the size, this membrane filter can filter all vegetative cells of microbes, because all the bacteria having the larger size of 0.2 micrometers. So by which this we can say that this mask cannot filter the virus, and we generally use in this for the protection against the viruses or the COVID viruses, but this mask cannot be sufficient to prevent the COVIDs. Another filter which is termed as HEPA, and we generally use in biosafety cabinet. Biosafety cabinets, or generally we use is laminar air flow. HEPA, the full form of this HEPA is high efficiency particulate air. And this filter has been installed in our biosafety cabinets, and by this we can achieve the microbes or the vegetative cells free air in our biosafety cabinets. So we can work over here. So this is the physical methods or the mechanical methods by which can control the microbes. In next section we will discuss about the chemicals by which we can control the microbes.